Okay, so we are starting our 5th unit on the course, called DNA and protein synthesis. But before I introduce the unit, I do want to mention that test corrections are available to do for our 4th unit test, which we took yesterday. And completing test corrections can potentially earn you up to half of the credit that you lost back. So we did try this out with unit 3. Not many of you did complete the test corrections, but this option is still available to you if you'd like to earn some credit back on your exams. You will need to do these test corrections by Friday. They are available on Canvas at the top of our Canvas page. And the directions are the same as last time. You do need to make sure that you do 2 things. Number 1, save your test correction form in your biology Google folder. And number 2, submit your test correction form to the assignment on Canvas. So save it in Google folder, submit it on Canvas, and you can earn potentially half of your missed credit back. All right. But we're introducing our 5th unit of the course on DNA and protein synthesis. Today's date is October 6th, 2020. It is Unit 5, Day 1. Our 5th unit is all about how DNA codes for proteins. And because of this, this process is often referred to as the central dogma of molecular biology, or the central dogma of biology, meaning it is the most pervasive and important theory in biology. What it says is that we can use DNA to code for specific proteins, and that DNA is also inheritable, meaning it can be passed on from one generation to the next. And it serves as the instruction manual to make the proteins that make life possible. We'll introduce this unit. As you all take out your notes, please make sure you have your notes out at all times. We are introducing this unit by first talking about the structure of DNA and how that structure determines its function. Okay, so another common theme in science is that structure determines function. If you change the shape of something, then you will ultimately change the job that that thing is able to do. Okay. We do have a midterm coming up for you all in this class. It will be next Wednesday, I believe. Wednesday? Next Wednesday. And we have about 15 different standards, 14 different standards that we need to cover. We've already covered 11 of them. Let me see if that's right math. Yeah, we've covered 11 of them. And then we've got a few more to cover as well before the exam. We probably will get through them all before the exam, and we will continue to review throughout this unit so that you all feel prepared for the midterm. Additionally, we will be making review videos for each of these standards so that if you're unclear about one of these topics, you can watch a quick 5-minute video that goes over the topic, pause it as necessary, rewind it as necessary, take notes as necessary, and feel more prepared for the midterm, which will take place next week. We don't want to put any more work on your plate. We know that there's a lot going on, not only in our biology class, but in other classes that you all are taking as well. So we don't want you to feel stressed out or discouraged. But this midterm is an opportunity for you to show what you know. It's not an opportunity for you to feel like we're just trying to make you feel stupid or like you haven't worked hard enough. It's an opportunity for you to show what you know. So between now and then, next week, you do want to make sure you're putting yourself in the best possible position to show that you know a lot, which you do. Okay. So here's a little bit of review from our first unit when we talked about the biological macromolecules. The question is, what are the subunits of DNA and their function? You all can either type it in the chat what you think the answer is, or someone can come off of mute and tell me what they think the answer is. Any thoughts about what this answer is? Thank you, Jaden. Well, you put in 2 different answers. Okay, so I will say one of the, that wasn't helpful. Anybody else have any thoughts? Again, the question is, what are the subunits of DNA? So what are the building blocks of DNA? We've got 4 options here. What are the building blocks of DNA? Okay. Nick and Alyssa, you are correct. The answer is A. So, guys, make sure you have your notes out here so you can refer back to things that you've hopefully written down in the past. That's why we write things down. It's not just to exercise our fingers, it is to make sure that we have a way to store information, just like DNA stores information. See what I did there? But we store information so that we can come back to it and refer to it and build upon it. So, in this case, the question was, what are the subunits of DNA and their function? The answer is nucleotides that store information. So DNA stands for deoxyribonucleic acid. We've got to start drawing the connection between nucleic acid and nucleotides and the nucleus. They're all referred to with the same general prefix for a very specific reason. They're all interconnected. So DNA is, of course, made up by nucleotides. Jaden, you said A, B, C, and D. So you don't get credit for just guessing all of the answer choices. All right. What else do we remember about DNA? We've learned some things in the last 4 units about DNA. What else do we remember about it? You can send it to the chat, or you can unmute yourself. What else do we remember? I'll give you credit, Jaden, if you can tell me one more thing that you remember about DNA. What else do we remember about DNA? Okay, so we remember good, Makaran. T, A, C, and G. Those are the 4 nitrogen bases. We also remember Jaden says that we are humans are 99.9% alike. That's also correct. Okay, we know that there is a connection to proteins. I wouldn't necessarily say that it's based off of proteins, but there is a connection. Okay. Well, hopefully we can start to think of some more things as we go, because we have talked about DNA quite a bit at this point, but we will retouch on some of those things just to provide a brief refresher. So this is when you should really start to write things down, especially if you're a person who does not have many notes from the past available to you from units 1 and 3 primarily, and 2. So DNA structure and function. DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid. DNA is a type of nucleic acid, meaning that its monomers are, as Alyssa told us, and who else? McChyron told us, the monomers are nucleotides. The structure of DNA is a double helix, so it looks like a ladder that has been twisted into a spiral. And the function of DNA is to store genetic information. We also know that all living things have DNA. That's what it says at the bottom of your screen there. All living things have DNA. So this is important to note. So hopefully you've been writing down these notes. Okay. So the DNA structure and function. So again, if I change the structure of DNA, then ultimately I am going to change the job that it's able to do. I'm going to change its function. Now, I want to be clear. When I say that I'm changing the structure of DNA, I'm not necessarily changing its shape. All DNA is shaped in the form of a double helix. It's shaped like a ladder that has been twisted into a spiral. When I say that I'm changing the structure of DNA, what I mean is I can rearrange the nitrogen bases so that instead of being in the order of C, A, G, T, maybe I reorder it so that it's A, G, T, or maybe G, A, G, T, or maybe C, A, T, T. That's what I mean when I say changing the structure of DNA. I'm changing the order of these nucleotides, of these nitrogen bases. Okay, so these nucleotides that make up DNA are made up themselves of three parts: the nitrogen base, which I just talked about, and as McChyron told us, can be either C, A, G, or T. There's a deoxyribose sugar. I know that's a long word, but of course that's where we get the word deoxyribonucleic. And then there's also a phosphate group. So those are the three parts of a nucleotide. When we string nucleotides together, we start to form nucleic acid or DNA. So nucleotides are made up of these three parts: nitrogen bases, deoxyribose sugar, and a phosphate group. Chain them together, we end up making nucleic acid. In this case, we've got DNA. There are 4 possible nitrogen bases: A for adenine, T for thymine, G for guanine, and C for cytosine. Hopefully you remember some of this from Unit 1, and that's why we're not having you write down everything you see, because it should already be in your Unit 1 notes. But this is good to know. All right. DNA is made up of a deoxyribose sugar and the phosphate backbone, as well as your nitrogen bases. This is what I was just talking about. So I'll leave this up for 10 seconds so you all can write it down, but it's essentially what I was just mentioning. All right. So here we can see this phosphate backbone again. The backbone is essentially the sides of the ladder. Remember, it's like DNA is like a ladder that has been twisted into a spiral. The sides of the ladder would be the sugar phosphate backbone. And then the rungs of the ladder, where you actually climb up the ladder, would be made up of your nitrogen bases: adenine, thymine, cytosine, and guanine. Those nitrogen bases, this is important, so please do write this down. The nitrogen bases are held together by weak hydrogen bonds. And those hydrogen bonds help to stabilize the DNA molecule. They help to keep it in a nice orderly shape. The nitrogen bases are also complementary, and this means that they get paired with one another. A will always be paired with T, T will always be paired with A, G will always be paired with C, C will always be paired with G. AT, CG. ATCG. Okay. So another practice question. Remember, we want to be thinking about those test taking strategies that we talked about on Friday, in which you try to highlight keywords, you eliminate any useless information, you make your brain dump, and then you make a prediction about what the answer will be. Okay, so our question here says or asks, how does our DNA make us unique? How does our DNA make us unique? So I want you all to send your answers to the chat. Do you think it's A, B, C, or D? Send your answers to the chat when you think you've got it. Okay, we've got a few answers coming in. Let's get some more answers coming in, folks. What do you think the answer to this question is? How does our DNA make us unique? Okay. Okay, so we've got B's and C's. The question, how does our DNA make us unique? Answer choice A says the sequence of nucleotide bases determine the proteins you produce and the combination of traits you express. Okay, so I just made a mistake because before you look at the answer choices, you really want to do your best to try to make a prediction about what the answer will be. So in this case, our question is all about DNA. So we should kind of think about what we know about DNA before we even look at the answer choices. So I know that DNA is a nucleic acid. I know that it's made up of nucleotides. And I know that those nucleotides have nitrogen bases like A and C and G and T. So based on what I already know about DNA, I should really be thinking about how those nitrogen bases contribute to the DNA making us unique. Okay, so that's how I can start to make a prediction about what the answer will be. Answer choice B says each person has a different amount of DNA in their cells, which determines the combination of traits they show. Answer choice C says the shape of each person's DNA is unique, making that individual show a unique combination of traits. And answer choice D says each person has different sugar phosphate backbones, which determines the combination of traits. All right. So they all at the end say determines the combination of traits. So that's not going to be the difference between these answer choices. The difference between these answer choices is going to come in the first part of the sentence. Let's look at B, since we had several people thinking that the answer was B. Each person has a different amount of DNA in their cells. Here's a simple question I can ask to prove to you that that is not correct. In our last 2 units ago, we talked about how many chromosomes humans have. Does anybody remember how many chromosomes humans have? What was that number? It was an even number. How many chromosomes do humans have? Remember, you get a certain number from your mom, you get that same number from your dad, and so together they add up to
23.
You get 23 from each parent. So how many do you have in total?
46.
46. Good. Yeah. So everybody has 46 chromosomes. So it's not the amount of DNA that's different. We all have the same amount of DNA. And all of your cells has the total package of DNA inside of it. So that's not really what we're thinking about here. A lot of people said C as well. The shape of each person's DNA is unique, making that individual show a unique combination of traits. So here's one thing that I love about the fact that you all chose this answer. You saw the word shape and you saw the word unique. And we just talked about the fact that if you change the shape, then you change the function. However, remember on this slide here, where is it? On this slide, I told you that it's not the shape that is changed. All DNA is in the shape of a double helix. All DNA looks like the ladder that has been twisted in the form of a spiral. Okay, so yes, the structure is changing, but it's not the overall shape. The overall shape is the double helix. So it's neither B nor C. Let me go back to the slide. The correct answer is A. It's the sequence of the nucleotide bases. Please write this down. The sequence of nucleotide bases determines the proteins you produce and the combination of traits you express. That's very, very important. The sequence of the nucleotide bases ultimately determines the traits. If I change the sequence, I more than likely will have an impact. Well, not more than likely, but it is possible that I will have an impact on the traits that are expressed. So very, very slight changes in our DNA between me and Jaden, or me and McKyron, or me and Ashley, or me and Jessica, all of these very slight changes produce all of the differences between all of us. They're very small changes, meaning instead of this being a C, then maybe I'll make it a T and see how that changes things. Instead of this being a T, then maybe I'll make it an A and see how that changes things. All right, so if you change the sequence of the nucleotide bases, then you have an impact. You can have an impact on the traits that are expressed. So please make sure you write that down. That's really, really important to note. Okay. Where is DNA found in cells? Does anybody know where we find DNA inside of a eukaryotic cell? Where do we find the DNA in a eukaryotic? What was that? Nonsense? Gibberish? Where can we find the DNA in a eukaryotic cell? Come on, think back to Unit 2 when we talked all about cells. Not in the mitochondria. No, not in the mitochondria. We don't find any DNA there. Where is the genetic information stored? Who's the boss of the cell?
Nucleus.
Thank you so much, Jaden. In eukaryotic cells, we find all of the DNA inside of the nucleus. Come on, guys. This is why I keep telling you, you got to remember that DNA, that's deoxyribonucleic acid, is made up of nucleotides and is found in the nucleus. Okay, we've got to keep that in mind. And then lastly, the second point here is DNA is found in the blank of prokaryotic cells. And literally the word nucleus was right there. I don't know what you guys were looking at. Where do we find DNA inside of a prokaryotic cell?
Plasmid.
Yes, it's in the plasmid, which just kind of swims around in the cytoplasm. Thank you. Not the cell membrane. So please, again, make sure you're taking these good notes, folks, because this is some information that we should hopefully remember going forward. Okay. So again, hopefully you wrote down that last answer choice, but we'll reiterate that it is the order of nitrogen bases that creates different proteins, which leads to different traits. So we can see here that we share huge, significant portions of our genes with chimps, significant portions of our genes with mice, almost half with flies, just about a quarter with yeast, and almost a fifth with plants. So there are very few genes that are totally unique to humans. All right. So genes code for proteins, which eventually lead to the traits. Proteins give us our traits like our skin color, our hair texture, our hair color, in some cases our height, in many cases the unfortunately some of the diseases that we are likely to develop over the course of our lifetime. So the genes are responsible for coding for all of that information. But genes are only 3% of your DNA. So a gene is just a section of DNA that codes for something. And only 3% of our DNA is genes. So the portion of our DNA, the vast majority of our DNA, is not genes, and it actually controls how the genes are produced and when they're produced and where they're produced. There are portions of our DNA that we simply don't fully understand at this point, that don't have an obvious job, but they still appear in our genetic code. So this is one of the more perplexing topics in science these days. Okay, so we've got about 28 minutes left in the class. At this point, you all are left to do your exit ticket. There is an asynchronous assignment called the Amoeba Sisters DNA Structure Video and Questions. Once you finish those two things, you can think about completing some missing work or doing some test corrections. The exit ticket, of course, shouldn't take you long at all. The Amoeba Sisters activity is pretty short as well. So go ahead and knock those out so you can stay on pace. And I will be here on mute. Okay, it's 1:20, folks. So you all are good to go now and enjoy the rest of your Tuesday. Please do not forget to stay up to date with your work and to finish the test if you have not done so up until this point. I will talk to you all tomorrow.